Glyceraldehyde 3-phosphate dehydrogenase, abbreviated as GAPDH or less commonly as G3pDH. EC 1.2.1.12 is an enzyme of approximately 37 kDa that catalyzes the sixth step of glycolysis and thus serves to break down glucose for energy and carbon molecules. In addition to this long-established metabolic function, GAPDH has recently been implicated in several non-metabolic processes, including transcription activation, initiation of apoptosis, to Golgi vesicle shuttling, and fast axonal, or axoplasmic transport. In sperm, a testis-specific isoenzyme GAPDHS is expressed. Structure. Under normal cellular conditions, cytoplasmic GAPDH exists primarily as a tetramer. This form is composed of four identical 37 kDa subunits containing a single catalytic thiol group, each critical to the enzyme's catalytic function. Nuclear GAPDH has increased isoelectric point pi of pH 8.3 to 8.7. Of note, the cysteine residue C152 in the enzyme's active site is required for the induction of apoptosis by oxidative stress. Notably, post-translational modifications of cytoplasmic GAPDH contribute to its functions outside of glycolysis. Interestingly, GAPDH is encoded by a single gene that produces a single mRNA transcript with no known splice variants, though an isoform does exist as a separate gene that is expressed only in spermatozoa. Reaction. Compound C00118 at KEGG pathway database. Enzyme 1.2.1.12 at KEGG pathway database. Reaction R01063 at KEGG pathway database. Compound C00236 at KEGG pathway database. Two-step conversion of G3p. The first reaction is the oxidation of glyceraldehyde 3-phosphate, G3p, at the position 1. In the diagram it is shown as the fourth carbon from glycolysis, in which an aldehyde is converted into a carboxylic acid. Delta G degree equals -50 kJ per mole, -12 kilocalories per mole, and NAD+ is simultaneously reduced endogonically to NADH. The energy released by this highly exergonic oxidation reaction drives the endogonic second reaction. Delta G degree equals +50 kJ per mole, +12 kilocalories per mole, in which a molecule of inorganic phosphate is transferred to the GAP intermediate to form a product with high phosphoryl transfer potential. 13-bisphosphoglycerate, 1,3-BPG. This is an example of phosphorylation coupled to oxidation, and the overall reaction is somewhat endogonic. Delta G degree equals +6.3 kJ per mole, +1.5. Energy coupling here is made possible by GAPDH. Mechanism. GAPDH uses covalent catalysis and general base catalysis to decrease the very large and positive activation energy of the second step of this reaction. First, a cysteine residue in the active site of GAPDH attacks the carbonyl group of GAP, creating a hemithioacetyl intermediate. Covalent catalysis. Next, an adjacent, tightly bound molecule of NAD+ accepts a hydride ion from GAP, forming NADH. GAP is concomitantly oxidized to a thioester intermediate using a molecule of water. This thioester species is much higher in energy than the carboxylic acid species that would result in the absence of GAPDH. The carboxylic acid species is so low in energy that the energy barrier for the second step of the reaction phosphorylation would be too high, and the reaction, therefore, too slow and equilibrium too unfavorable for a living organism. Donation of the hydride ion by the hemethioacetal is facilitated by its deprotonation by a histidine residue in the enzyme's active site. General base catalysis. Deprotonation encourages the reformation of the carbonyl group in the thioester intermediate and ejection of the hydride ion. NADH leaves the active site and is replaced by another molecule of NAD+, the positive charge of which stabilizes the negatively charged carbonyl oxygen in the transition state of the next and ultimate step. Finally, a molecule of inorganic phosphate attacks the thioester and forms a tetrahedral intermediate, which then collapses to release 13-bisphosphoglycerate and the thiol group of the enzyme's cysteine residue. Regulation. This protein may use the Morphian model of allosteric regulation. Function. Metabolic. As its name indicates, glyceraldehyde 3-phosphate dehydrogenase, GAPDH, catalyzes the conversion of glyceraldehyde 3-phosphate to deglycerate 13-bisphosphate. This is the sixth step in the glycolytic breakdown of glucose, an important pathway of energy and carbon molecule supply which takes place in the cytosol of eukaryotic cells. The conversion occurs in two coupled steps. The first is favorable and allows the second unfavorable step to occur. Transcription and apoptosis. GAPDH can itself activate transcription. The OCA-S transcriptional coactivator complex contains GAPDH and lactate dehydrogenase, two proteins previously only thought to be involved in metabolism. GAPDH moves between the cytosol and the nucleus and may thus link the metabolic state to John transcription. In 2005, Hara et al. Showed that GAPDH initiates apoptosis. This is not a third function, but can be seen as an activity mediated by GAPDH binding to DNA like in transcription activation. Discussed above, the study demonstrated that GAPDH is S-nitrosylated by NO in response to cell stress, which causes it to bind to the protein SIAH1. A ubiquitin ligase. The complex moves into the nucleus where SIA1 targets nuclear proteins for degradation, thus initiating controlled cell shutdown. In subsequent study the group demonstrated that deprenyl, which has been used clinically to treat Parkinson's disease, strongly reduces the apoptotic action of GAPDH by preventing its S-nitrosylation and might thus be used as a drug. Metabolic switch. GAPDH acts as reversible metabolic switch under oxidative stress. When cells are exposed to oxidants, they need excessive amounts of the antioxidant cofactor NADPH. In the cytosol, NADPH is reduced from NADP+ by several enzymes. Three of them catalyze the first steps of the pentose phosphate pathway. Oxidant treatments cause an inactivation of GAPDH. This inactivation reroutes temporally the metabolic flux from glycolysis to the pentose phosphate pathway, allowing the cell to generate more NADPH. Under stress conditions, NADPH is needed by some antioxidant systems including glutaredoxin and thioredoxin, as well as being essential for the recycling of glutathione to Golgi Transport. GAPDH also appears to be involved in the vesicle transport from the endoplasmic reticulum to the Golgi apparatus, which is part of shipping route for secreted proteins. It was found that GAPDH is recruited by RAB2 to the vesicular tubular clusters of the where it helps to form COP1 vesicles. GAPDH is activated via tyrosine phosphorylation by SRC. Additional functions. GAPDH, like many other enzymes, has multiple functions. In addition to catalyzing the sixth step of glycolysis, recent evidence implicates GAPDH in other cellular processes. GAPDH has been described to exhibit higher-order multifunctionality in the context of maintaining cellular ion homeostasis. This came as a surprise to researchers, but it makes evolutionary sense to reuse and adapt existing proteins instead of evolving a novel protein from scratch. Use as loading control. Because the GAPDH gene is often stably and constitutively expressed at high levels in most tissues and cells, it is considered a housekeeping job. For this reason, GAPDH is commonly used by biological researchers as a loading control for Western blot and as a control for qPCR. However, researchers have reported different regulation of GAPDH under specific conditions. For example, the transcription factor MZF1 has been shown to regulate the GAPDH gene. Therefore, the use of GAPDH as loading control has to be considered carefully. Cellular distribution. All steps of glycolysis take place in the cytosol, and so does the reaction catalyzed by GAPDH. In red blood cells, GAPDH and several other glycolytic enzymes assemble in complexes on the inside of the cell membrane. The process appears to be regulated by phosphorylation and oxygenation. Bringing several glycolytic enzymes close to each other is expected to greatly increase the overall speed of glucose breakdown. Recent studies have also revealed that GAPDH is expressed in an ion-dependent fashion on the exterior of the cell membrane, where it plays a role in maintenance of cellular ion homeostasis. Clinical significance. Cancer. GAPDH is overexpressed in multiple human cancers, such as cutaneous melanoma, and its expression is positively correlated with tumor progression. Its glycolytic and anti-apoptotic functions contribute to proliferation and protection of tumor cells, promoting tumorigenesis. Notably, GAPDH protects against telomere shortening induced by chemotherapeutic drugs that stimulate the sphingolipid ceramide. Meanwhile, conditions like oxidative stress impair GAPDH function, leading to cellular aging and death. Moreover, depletion of GAPDH has managed to induce senescence in tumor cells, thus presenting a novel therapeutic strategy for controlling tumor growth. Neurodegeneration. GAPDH has been implicated in several neurodegenerative diseases and disorders, largely through interactions with other proteins specific to that disease or disorder. These interactions may affect not only energy metabolism but also other GAPDH functions. For example, GAPDH interactions with beta-amyloid precursor protein, beta-ap, could interfere with its function regarding the cytoskeleton and membrane transport, while interactions with Huntington could interfere with its function regarding apoptosis, nuclear tRNA transport, DNA replication, and DNA repair. In addition, nuclear translocation of GAPDH has been reported in Parkinson's disease, PD, and several anti-apoptotic PD drugs, such as rasagiline, function by preventing the nuclear translocation of GAPDH. It is proposed that hypometabolism may be one contributor to PD, but the exact mechanisms underlying GAPDH involvement in neurodegenerative disease remains to be clarified. The SNPRS 3,741,916 in the 5' UTR of the GAPDH gene may be associated with late-onset Alzheimer's disease. Interactions. Protein-binding partners. GAPDH participates in a number of biological functions through its protein-protein interactions with nucleic acid binding partners. GAPDH binds to single-stranded RNA and DNA, and a number of nucleic acid binding partners have been identified. Inhibitors. Cholinergic acid. Interactive pathway map. Click on genes, proteins and metabolites below to link to respective articles. Further reading.